Thank you for inviting me as a speaker. Our laboratory studies brain development, including development of a hippocampus, which plays an essential role in learning, memory, and spatial navigation. This picture shows a coronal section of an adult mouse hippocampus. You can see what hippocampus consists of the dentate gyrus, brain regions where neurogenesis occurs throughout the life. And hippocampus also contains CA1, CA2, and CA3 regions. Dentate gyrus is segregated from the rest of the hippocampus by a groove called hippocampal fissure. One approach to study hippocampal development is to analyze mouse mutants. We studied mice in which limb homeodomain transcription factor Lmx1a is inactivated by a point mutation, and we observed hippocampal abnormalities in these mice. So these are coronal sections of adult mouse hippocampus, and you can see dentate gyrus and CA1, CA3 regions in wild-type mice, and in Lmx1a mutants, CA1, CA3 regions were slightly reduced, but the dentate gyrus and hippocampal fissure were dramatically reduced. To better analyze dentate gyrus neurons, we used PROX1 immunohistochemistry. PROX1 is specifically expressed in dentate gyrus neurons. And we observed what, in contrast to wild-type mice, in Lmx1a mutants, there were much fewer dentate gyrus neurons, and this difference was very statistically significant. We also studied input resistance of individual dentate gyrus neurons in wild-type and Lmx1a mutants. Input resistance depends on neuronal morphology and the number of ion channels. And we observed what compared to wild-type controls, input resistance in dentate gyrus was significantly reduced in Lmx1a mutants, suggesting abnormal function of dentate gyrus in these mice. So, to identify developmental origin of these abnormalities in Lmx1a mutants, we analyzed early stages, beginning with postnatal day 3, P3, and images from this developmental stage are shown here. So first, we used CTIP immunohistochemistry to study general hippocampal pattern. CTIP labels dentate gyrus neurons and also neurons in CA1 and CA2 regions. But neurons in CA3 region are negative for the CTIP stain. So the same pattern was observed in Lmx1a mutant hippocampus, indicating that no differences in hippocampal patterning occur in these mutants. When we analyzed the number of dentate gyrus neurons with PROX1 immunohistochemistry at this developmental stage, and already observed a significant reduction in the number of dentate gyrus neurons, indicating what abnormal development earlier than postnatal 3 probably contributes to hippocampal abnormalities in Lmx1a mutants. So during development, and we investigated embryonic stages. During development, dentate gyrus neural progenitors originate in the dental neuroepithelium, labeled as DNE here, in the telencephalic ventricular zone. So these neurons exit dentate neuroepithelium at embryonic day 14 in the mouse. They first migrate radially and then extend along the outer surface of the telencephalon for a while. and then migrate again radially along the hippocampal fissure to form the dentate gyrus. To study migration of dentate progenitors, we used BRDU labeling. BRDU is incorporated into the DNA of dividing cells, and once it is incorporated, it's permanently present in DNA of such cells, and these cells can be identified using immunohistochemistry against BRDU. So once injected into mice, BRDU is present for approximately 24 hours, and all proliferating cells in this time frame become labeled with BRDU. So, and we can follow up with cells at later time. So we injected mice with BRDU at embryonic day 14.5 and analyzed them at embryonic day 18.5, 4 days later. So here is BRDU immunohistochemistry in wild-type and Lmx1a mutant mice. So cells, progenitors from dentate neuroepithelium migrate first into migrate into a dentate gyrus through dentate migratory stream. So first segment of their migratory route is called dentate migratory stream here. Then they migrate along fimbrial dentate junction here, and then finally enter the dentate gyrus. So you can see what in wild-type mice, a significant portion of BRDU positive progenitors end up at this point at the dental migratory stream. Many of them reach FDJ, and some already reach the dentate gyrus. But in Lmx1a mutants, you see most of them are located in the DMS segment of a migratory stream, in the first segment of a migratory stream. Then very few of them reached the FDJ, and few of them reached the dentate gyrus. And this difference is statistically significant. So many cells in Lmx1a mutants are still in the DMS, fewer in FDJ, and fewer in the dentate gyrus compared to wild-type controls, indicating abnormal migration of dentate progenitors in Lmx1a mutants. So during the migration to the dentate gyrus, progenitors migrate along glial fibers. So we identified glial fibers using anti-GFAP immunohistochemistry. So these pictures at the bottom correspond to boxed regions in the top panels. So we analyzed GFAP staining at embryonic day 18.5 in wild-type and Lmx1a mutant mice. You can see numerous glial fibers in the developing hippocampus of wild-type embryos. But in Lmx1a mutants, the number of glial fibers was dramatically reduced. They were almost gone, suggesting that loss of Lmx1a prevents the development of glial scaffold, and this in turn may affect migration of progenitors. So when we studied a proliferation of dentate progenitors in the ventricular zone, so dentate neuroepithelium was identified using PROX1 immunohistochemistry, and regions boxed in the upper panels are shown at high magnification at the bottom. So again, we used BRDU to identify proliferating cells. Cells with dividing cells incorporate BRDU into their DNA and can be identified based on immunohistochemistry against BRDU. So DEPI stains all nuclei, and BRDU stains nuclei of proliferating cells. We observed a dramatic reduction in the proliferation in dentate neuroepithelium in Lmx1a mutants, and this difference was statistically significant. So we conclude that in addition to glial scaffold abnormalities and migration abnormalities, loss of Lmx1a reduces proliferation in in the dentate neuroepithelium. So to understand the function of Lmx1a during hippocampus development, we next studied expression pattern for Lmx1a. These are coronal sections from embryonic day 12.5 and 18.5 mouse wild-type embryos, and we used anti-Lmx1a antibody. And interestingly, we did not observe Lmx1a expression in the hippocampus at all. Instead, Lmx1a expression was observed adjacent to the hippocampus, in particular in the cortical hem, and also in the choroid plexus epithelium here. And cortical hem is interesting because it is a signaling center which non-autonomously regulates hippocampus development. Cortical hem produces secreted factors such as Wnt3a, which promote specification and development of a hippocampus. So, and as development proceeds, a cortical hem becomes fimbria. And you can see what Lmx1a is expressed in the cortical hem, and then its expression is maintained in fimbria at later stages, but no expression of Lmx1a is present in the hippocampus itself. So we hypothesized that Lmx1a regulates expression of Wnt3a in the cortical hem. And we studied Wnt3 expression in wild-type embryos and Lmx1a mutants using in situ hybridization. You can see what Wnt3 is strongly expressed in wild-type cortical hem, but its expression is dramatically reduced in Lmx1a mutants. And this difference was statistically significant. So Lmx1a indeed regulates Wnt3 expression in the cortical hem. To further test what Lmx1a functions via Wnt3a, we performed analysis of double gene heterozygous mice. So we analyzed mice at postnatal day 3 and counted the number of the PROX1 positive dentate gyrus neurons in wild-type mice and single gene and double gene heads. So we observed similar number of dentate gyrus neurons in wild-type, Lmx1a heterozygous mice, Wnt3 heterozygous mice, but a dramatic reduction of dentate gyrus neurons was observed in Lmx1a, Wnt3 double heterozygous mice. This difference was statistically significant, indicating that Lmx1a and Wnt3 indeed act in the same pathway to regulate dentate gyrus development, in particular the number of dentate gyrus neurons. When we studied input resistance in Lmx1a Wnt3a single and double head mice, and there was no difference in input resistance between single head mice and wild-type mice, but there was a dramatic reduction in the input resistance in Lmx1a Wnt3a double head mice. Next, we studied a glial scaffold in the same mice. So, as before, glial fibers were identified based on GFAP immunohistochemistry. You can see glial fibers here in wild-type embryos, but in Lmx1a Wnt3a double heads, the number of glial fibers was dramatically reduced, and this difference was statistically significant. So, and finally, we studied proliferation in the dentate neuroepithelium. Again, we used PRTU. Mice were injected at embryonic day 14.5 and analyzed 90 minutes later. So proliferating cells are green here, and we observed a dramatically reduced number of proliferating cells, a reduced fraction of proliferative cells in the dentate neuroepithelium of Lmx1a Wnt3a double head mice compared to wild-type mice and single head mice. So we conclude that Lmx1a is required for development of dentate gyrus by regulating proliferation and migration of dentate gyrus progenitors, and also by regulating glial scaffold formation. And second, we identified that Lmx1a regulates dentate gyrus development via expression of secreted Wnt3a in the cortical hem signaling center. I'm grateful to members of our laboratory, Igor Iskusnik and Nikolai Fatahov, who performed most of experiments what I showed today. And I'm also grateful to our collaborators from the University of Tennessee Health Science Center and from St. Jude Children's Hospital. Thank you for attention.